Good evening, everyone. Just confirm whether the audio and the video is clear to you or not. Today we'll be discussing few more things related to the cell signaling unit. If you have visited like the previous class that we had yesterday, so we had a discussion regarding what is cell signaling all about and its various types, and in today's class we'll be dealing with few other topics related to the same unit. Let's wait for the students to join in and then we'll begin with our class. Till then, just confirm the students who are there in the class whether the audio and the video is clear to you or not. Just confirm, everyone. Yes, we have 4 students in the class. Just confirm quickly whether it is clear or not. Okay, Janvi, thank you. Okay, so let's begin. Okay, thank you for confirming. First of all, if you haven't yet joined us on our application, make sure that you download the application of Botany Insider, both from the Play Store and also from the iOS. It is available both for the Android users and also for the iOS users. We have a variety of courses available for you all. You can enroll in that. And the very important, very fresh course that is running for the time being is this particular one, which is Target the June 2023 course. It is a full course with all the theory lectures, general aptitude classes, hard copy notes, PYQ sessions, doubt sessions. Every single thing would be provided to you in the same evening, Kaveri. So this was about it. In addition to that, we have the hard copy notes now available for you all. Doorstep delivery now available. Make sure that you order this via the WhatsApp number that is given to you in the description box. In addition to that, you can place your orders via the application as well. And also the mock test series is now available for the June 2023, in which we have 40 plus mock tests within all India ranking analysis and all the other important contents for the same. Okay, now yesterday we discussed about what is cell signaling and its type. So today we'll be understanding about the GPCR, that is the G protein coupled receptor signaling. So these classes are specially designed for one specific student, I must say, because she was insisting upon that she wanted to learn about this, so I thought of to help her out. So I hope that person is getting the help for these things, and all the other students would be getting the benefit, because cell signaling is a unit which is very, very important. First and second, it is a bit dicey and bit complicated for some students, so I thought of taking up the same thing. Okay, so talking about GPCR, that is G protein coupled receptor. First thing first, this particular topic, if you look into the CSIR's syllabus, if you look into the previous year questions, you will see every single time we find a question from this particular topic. Okay, so be attentive in the class. Make sure that you are following each and every point. Very first thing, what is GPCR? Look into the structure first. So these are all my handwritten notes. Okay, and in the same manner we go for every single thing in the structured courses, where we do all the signaling, every single topic via my notes itself. So these are my notes which I made during my preparation. So I have clicked the picture and uploaded the same thing, so it would be much more easier for you to understand. So GPCR, it is G protein coupled receptor pathway. So in this, what happens, we have a G protein utilized, we have a G protein which helps in the complete pathway. It is serpentine in nature. What do you mean by serpentine? सर्पेंटाइन का सिंपल मतलब होता है कि इसके अंदर 7 ट्रांस मेम्ब्रेन लूप्स होते हैं. नाउ इफ यू लुक इनटू द स्ट्रक्चर ओवर हियर, काउंट 1 2 3 4 5 6 7. सो इन टोटल वी हैव 7 ट्रांस मेम्ब्रेन स्ट्रक्चर्स प्रेजेंट. ये सारे जो लूप्स हैं ये 7 टाइम हमारी प्लाज्मा मेम्ब्रेन को क्रॉस करेंगे और यहाँ पर 3 लूप्स बनेंगे. इफ यू लुक क्लोजली, लूप 1, लूप 2 एंड लूप 3 जो लॉन्गर होता है. एंड अगर हम बाहर देखें तो जो एक्सटर्नल साइड पर है वहाँ पर हमारे पास ट्रांस मेम्ब्रेन्स होते हैं, ट्रांस मेम्ब्रेन लूप्स. सो ट्रांस मेम्ब्रेन 1 लूप, ट्रांस मेम्ब्रेन 2 लूप एंड ट्रांस मेम्ब्रेन 3 लूप. ओके, नाउ इट इज अल्फा हेलिकल स्ट्रक्चर. व्हाट इज अल्फा हेलिकल स्ट्रक्चर? सो अगर हम प्रोटीन के स्ट्रक्चर्स की बात करें, प्रोटीन के स्ट्रक्चर के अंदर वी हैव प्राइमरी स्ट्रक्चर PCR, वी हैव अल्फा हेलिकल स्ट्रक्चर्स प्रेजेंट. ओके, रिमेंबर अबाउट दिस, एवरी सिंगल पॉइंट रिटन ओवर हियर इज important. Okay, next, when you have a ligand, yesterday if you remember we have discussed about that what is the ligand, what is a receptor. Okay, so you have a ligand, you have a receptor. So what was a ligand? Ligand was the molecule that was produced by the inducer cell, right? And what was a receptor? So receptor was the structure that was present on the responder or the receiver's signal or the receiver's cell. So this ligand would come and bind over here. So in this particular thing, in this particular GPCR, the ligand comes and binds to the 6th and 7th loop. So this is the 6th and 7th loop where you have the ligand binding site. All these questions are asked. Make sure you are paying attention to these. And this third loop is attached to our G protein. That is the main protein which would be like actually working in the complete functionality. Okay, now if you look into the structure of the G protein, just a second. Yeah, now if you look into the structure of the G protein, so this G protein have 3 of the sites. It have 3 of the structures over here, alpha, beta. Look into the image over here, you can see alpha, beta and gamma is there. Now this is the main thing that you need to remember. Now let's understand about, or let's go through the main pointers. Very first point is GPCR. It binds with the ligand and activates some membrane protein called as G protein. So we have a G protein which gets activated via what? Via a ligand getting binded to the membrane protein. Now this activated G protein, it interacts with the enzymes in the membrane. So initially when alpha, beta and gamma, all the 3 are attached to our or they are in the close association forming the main G protein, it is inactive. But जैसे ही ligand आकर bind करेगा तो हमारा G protein जो है वो activate हो जाएगा और it would start to function. Now see, as and when the G protein gets activated, वो अपनी shape को change करेगा, active होगा और split कर जाएगा 2 different subunits के अंदर. One or both the subunits may be able to activate the other protein as a result. So जैसे ही G protein, because see this GPCR is the what do you say, basic protein that is helping. Now depending upon complete pathway it is functioning upon, वो अलग अलग proteins को जाके activate करेगा. ठीक है, so one or both the subunits may be able to activate the other protein as a result. So this GPCR, it is what? Remember this particular point as well, and that is GPCR is a guanine nucleotide binding protein. So what is it? It is guanine nucleotide binding protein which is acting as a molecular switch inside a cell, and they are involved in transmitting the signals from outside to the interior. Now important point is that they belong to a large group of enzymes called as the GTPase, which causes the GTP hydrolysis So see, look over here. Initially what happens when the GPCR is in inactive state? So it gets bounded to the GDP. But जब GDP GTP में convert होगा तब हमारा G प्रोटीन G अल्फा जो सब यूनिट है वो एक्टिवेट होगा, वो रिमूव आउट हो जाएगा अपने तीनों सब यूनिट्स के कंपोनेंट से. वहाँ पर अब पहले हमारे पास वी हैड अल्फा बीटा एंड गामा प्रेजेंट और ये जीडीपी से बाउंडेड था. यहाँ पर ग्वानाइन एक्सचेंज फैक्टर आएगा जो वर्क करेगा. वो कब आएगा? जब हमारे जी प्रोटीन के ऊपर लिगेंड ने आकर बाइंड किया. लिगेंड जो है वो इस प्रोटीन को एक्टिवेट करेगा, जी प्रोटीन को, जिसकी वजह से ग्वानाइन एक्सचेंज फैक्टर वुड कम एंड इट वुड एक्टिवेट द जी प्रोटीन, जिसकी वजह से जो अल्फा सब यूनिट है जी प्रोटीन की वो डिसोसिएट होगी कंप्लीट ओरिएंटेशन से और यहाँ पर GTP से वो बाउंडेड है. सो एज एंड व्हेन दिस गेट्स एक्टिवेटेड, अब क्या होगा? यहाँ पर वी वुड बी हैविंग आर एडिनाइलेट साइक्लेज एंजाइम गेटिंग एक्टिवेटेड. सो व्हाट विल दिस एडिनाइलेट साइक्लेज एंजाइम डू? इसकी वजह से हमारा जो ATP है, जो हमारा मेन सब यूनिट है, there is network issue. There must be some network issue from your end. Please check. Other students also, like, are you also facing the same problem or is it only with Janvi? Let me know, please. From my end, I guess it is working fine. Janvi, please check it from your end. I guess all the other students are able to watch it properly. Okay. Janvi, just check it from your end. I guess there is some... Okay, buffering is on your end as well. I checked it. What should we do? Let's wait for a minute, maybe it might get done. My Wi-Fi is working properly, OBS is working properly, everything is going well, stream health is perfect. Let's continue in the middle, we'll see. Okay, yeah, so we were discussing this G alpha that is the alpha subunit of the G protein would get detached and it would activate the adenylate cyclase. This adenylate cyclase would be activating the GTP, the ATP, and it would convert it into CAMP. Now this CAMP, see, for converting the ATP into the CAMP, we require adenylate cyclase, and for converting the CAMP back into ATP, this PDE is required, that is phosphate esterase. Remember about all these things, they are all very important. Okay, now these 4 CAMP molecules, they would activate the pKa, that is protein kinase A. So it would activate, which is initially inactive, it would come and bind, the 4 CAMP would come and they would activate the pKa. Now as and when this gets activated, this complete work was happening in the cytosol. Okay, now the cytosol would be, from this cytosol, this active pKa would enter into the nucleus. It would activate the CREB, that is the camp response element binding protein, from the inactive stage to the active state. Now the CREB plus CBP, that is the CREB binding protein, they both would come, join together with each other and they'll form CRE, that is camp response element. Now the CRE would be leading to the activation of the transcription factor, and finally we'll see the response of the associated pathway. Okay. Clear to everyone? Is there any doubt till here? Or is the Wi-Fi not working properly? Yes, Kaveri, just confirm whether it is working well or not now, because I can see the students leaving the class. Is there a network issue still? Because from my end I can see that everything is working fine. Okay, from your side it's fine. Okay, then it's absolutely correct then. Okay, so this was about the main concept, this was about the main functionality of how the GPCR works. Okay, now let's move on to the various other points. Okay, so talking about the secondary messenger, so these operate in the GPCR pathway to further generate the specific desired response after the ligand receptor interaction. So what are the secondary messengers? So if you look here closely, over here we saw that CAMP was getting activated. So these all are the secondary messengers. What was the initial messenger? The ligand that came over here to activate the G protein. That was the primary messenger. So when the ligand came, this would be binding, this would be activating the G protein, and on the activation what would happen? It would lead to the activation of the secondary messenger. What we understood was for the CAMP. In addition to that, there could be other ones as well, like CGMP, IP3, calcium 2+, or the triglycerol. Depending upon whatever desired response need to be there, the secondary messenger could change. So talking about the CAMP first, it is a derivative of ATP and it is used for the interaction for the intracellular transduction in the different organisms in the CAMP dependent pathways. Now its level is maintained via what? Level is maintained via the balanced regulation between adenyl cyclase and PDE. So we have just understood what is the correlation between the two. So ATP can get converted into CAMP via the adenyl cyclase, and CAMP gets converted into the ATP via the PDE. So via regulating the levels of adenylate cyclase and PDE, we can change, or we can actually control the levels or maintenance of the ATP and CAMP levels could be done. So it is synthesized from the ATP by adenyl cyclase, which is located on the inner side of the plasma membrane. Again, very important, where is it CAMP located? CAMP is located on the inner side of the plasma membrane. What are the significance of the CAMP? So the significance, very first one is it converts glycogen to glucose. It promotes the muscle contraction in the heart by increasing the heartbeat. It has a role in the absorption of water in kidneys and maintenance of memory in brain. So remember about all of these things, because in examination, in CSIR, in the GATE exam, which is very close, exam is on the second week of February. In the second week of February, we are about to have those admit cards. So remember about all these things, whatever is written on the presentation, all these things are important. Okay, so once the CAMP is activated, pKa, and if the pathway after the completion of the function is not regulated, it can lead to hyperproliferation and cancer. What does this mean? That means if we have like in the normal case as well, if there is a specific pathway occurring and if we are not regulating it, or if it is not regulated on a normal basis, what will happen? It could lead to hyperproliferation, right? The cells could get hyperproliferated, and on hyperproliferation it could lead to cancer as well. Because what is cancer? Cancer is uncontrolled division of cell, and no matter which of the component gets remains unregulated, it could lead to a major damage to our cells. So it could the non-regulation of this particular pathway as well could lead to hyperproliferation and cancer. Okay, now we have protein kinase, we have protein phosphatase and protein diesterase. Protein kinase is the one that transfers the phosphate on serine or threonine residues. Then we have protein phosphatase, that removal of the phosphate groups can activate or deactivate an enzyme. And protein diesterases can convert the cyclic GMP to 5-GMP. Okay, so these are the important points. Look over here. You have the GPCR, initially alpha, beta, gamma is there. Now it gets activated, G alpha would be there. It would either cause GS, GI or GQ depending upon the functionality. So GS would lead and GQ would lead to increase in the adenyl cyclase and PIPC. In the first one it could lead to CAMP increase of the CAMP pathway. Second could lead to decrease, and third could lead to the increase in the Ca2 plus ion. So this is related to the IP3 DAG pathway, which we might take up in the further classes. I'll plan and let you know, and as and when, depending upon the final scenario, there could be changes in the cytoskeleton or the morphology or the adhesion, and it could lead to increase or decrease in the impedance in sense of the cell functionality. Okay, are these things clear to everyone? Yes or no? We have 3 students in the class. Is everything clear to everyone? Clear everyone? Any doubt? Great. So this was a small topic that I wanted to discuss in today's class. Tomorrow we'll be having another marathon. We are taking small chunks of the topics every day so that you can come over to the classes and keep a track of the topics and prepare very nicely. Okay, so this was about today's class. Make sure that you download our application. We have a lot of content available for you all. Most importantly, we have the hard copy delivery, like the we have the notes booklet available for you all. If you wish, you can place the orders all over, like in complete India. You can get your notes delivered on your doorstep. You can WhatsApp on the number 9718170165. Okay, make sure if you wish to place your orders, you can place your orders. And also we have a course running for the target 2023 examination. It's a full theory course, 3 months course. We'll be done with the complete course by the end of March or in the first week of April. If you wish, you can enroll in that. Live and recorded lectures. Every day we are having 1.5 hours to 2 hours of teaching live on our application itself. So download the application and get yourself enrolled. Okay, thank you for joining in. Do not forget to get your notes booklet prepared, get your notes booklet delivered and ordered from our end. Okay, thank you for joining in. Let's meet tomorrow. Bye everyone. And for getting your notes, you can WhatsApp us on the number which is given to you in the description box. Okay, bye everyone, take care.